Yeah. Okay. Great. So my name is Vivek Kumar. I actually consider Memphis my hometown, so I grew up here. Great to be back. I was here last year, and I'm going to talk to you about the work in our lab using advanced behavior quantification. So I'll skip. Oh, okay. Got it. Let me skip the. Folks that did the work, but we're pretty much a multidisciplinary team. We have engineers and computer scientists working next to behaviorists and neuroscientists. And our overall goal in the lab is to really solve this problem of better clinical treatments for neuropsychiatric conditions. I think we can all agree that neuropsychiatry is a field where we're still using drugs with lots of side effects that are not optimal, and there really hasn't been a lot of innovation in treatment in terms of pharmaceuticals. And we believe that a big bottleneck here is the lack of really good animal models for neuropsychiatric traits, because animal models are really critical for any drug development efforts. And I'll skip a lot of the work and kind of talk to you more about the way we kind of think of the problem here. So our simple model here is I'm a geneticist, so I think of everything in terms of genetic circuits, and altered genetic circuits shape altered nervous systems, and then these altered nervous systems produced altered behavior. Most of us in this room are really good at 2 of these 3 layers, and we should all be good at all 3 layers. So my lab can do genetics at scale, and we can do behavior at scale. We kind of just bypass the nervous system. Many labs are really good at looking at nervous system activity and linking it to behavior. And ideally, what we'd like to do is kind of work at all 3 levels. And I'm going to show you how we can start to think about this problem at scale. So traditionally, the way animals, how we study behaviors in animals, if we could ask animal how it feels, we wouldn't really have much of a problem. But we can't, so we have to derive animals' emotional state or internal states through a series of tests. So here are some tests that are, social interaction tests. These are your state-of-the-art depression-like tests where you look at animals swimming or force them to swim or hang them upside down. And I think we can all agree these tests have low reproducibility, low throughput, and the validity of some of these tests come from the fact that if you give an animal a drug like an antidepressant before you throw them in a bucket of water, they'll struggle a little bit longer. So that assay becomes a depression-like assay, which I think is really wrong, because you're looking under the spotlight. The drug is causing 1,001 effects, and you're picking the one that you think it matters. So sure enough, drugs are developed or preclinical tests are done, and then we end up kind of, blaming the animal for all the problems of why things don't translate. So the question then my lab is really focused on, is there a better way? And a way, this idea has been put forward of looking at ethology or naturalistic behavior using computational tools, and we're really invested in this type of approach. So what is ethology? Ethology is essentially studying naturalistic behavior, hopefully in nature. This is Jane Goodall and studying her chimpanzees. One of her seminal kind of discoveries was that she looked at grooming behavior in these chimpanzees, and she showed that grooming is not just about keeping the fur coat clean, it's really about hierarchy. And the older animals will groom the younger animals will groom the older animals. Animals that have been kicked out of the group will groom the right individuals to get back into the group. The idea is that by studying this important behavior, you really have a completely different underlying causality to that behavior, which you find when you look at them in nature. So the idea here with computational ethology is to use computer vision and machine learning to automate the task of observing the animal. And this leads to reproducibility and replicability increases. We essentially have these standardized pipelines, and we have objective measures for scoring the behavior. A big bonus part of video data, which is used a lot for this work, is we can reuse the data continuously for different purposes. One person may look for grooming, the other one person may look for gait, so on and so forth. This is scalable. Scalability here, I think the limits are really how much compute you have, and how much access to models you have. And then hopefully, and this is a hope here, that there's increased translatability, i.e., the assays are going to give us more predictive power of what happens to a drug or a circuit perturbation in humans than we currently have with these artificial tests. Okay, so I'm at the Jackson Laboratory. We're mouse-centric people. And so what does ethology look like in the mouse? So this is a social behavior experiment that was done in 1955 in mice. This is an arena that's around 17 feet in diameter, or sorry, 14 feet in diameter. And the researchers introduced 56 mice into this arena, 28 males and 28 females. And then they watched the evolution of this colony over the next several weeks. We're talking 2-3 months here. And what they noticed was that the mice formed these kind of colonies. So here's one male and 2 female, and this is their territory. The males and the female will actively defend this territory. You have other kind of territories here. Majority of the animals are actually not in these territories. The majority of the animals are actually here. There are 8 males here in this one nest, and there are 13 males and 19 females in this one nest. These 8 males are actually defeated males. They're so defeated that what they have done is they've switched their circadian phase, and they'll go out looking for food and water during the daytime. So they don't have to interact with these guys here because, again, territoriality. These individuals here are dominated by one really aggressive male. And the term that the researcher used was to describe this male was that he terrorizes everyone else in this arena. Okay, so if I'm really interested in true social interaction of how dominance occurs, what's healthy association versus unhealthy association, I would study these individuals up here and compare their neural and genetic circuits and their behaviors to these individuals here. Of course, this is a really hard experiment to do. First, we're not going to get this large arena or 20 of them built in our labs. But the harder part here is how to score all these behaviors. Like, it's really hard to sit there and watch videos, so this is the part. These are the types of experiments we need to start to think about to do in the laboratory, but do it in an automated, scalable way that we can use in circuit perturbation studies, genetic and neural circuits, as well as drug discovery studies. Okay, so then the question is, how do we study naturalistic behaviors in lab animals? So I'm going to focus the next section of the talk on this and the work my lab has done. And we can think about studying naturalistic behaviors or unforced behaviors in kind of a couple of environments. So, and these are just stock photos I took off the internet, but in one environment, we can look at natural behaviors in mice in their homes, i.e., whatever the environment the mouse lives in. Let's put a camera in there and we watch the mouse 24/7 or some sort of sensor in their homes. So this is one approach. The advantage of this is a long-term, it's high throughput if you can get enough cameras and if you have enough computing power. It's lower resolution, meaning the amount of information you get about what the mouse is doing is probably not going to be as rich, mainly because the mouse can't do that much. And also it's a very enclosed environment. So sensors like cameras always will have trouble. But I'm going to give you an example of that in the second half of my talk. The first half of my talk I'm going to spend over here. And this is what I call kind of a hotel. So the mice come in, it's all fancy. There's lots of stuff for people to do, lots more space. And now you can actually get a lot more information about what the mouse is doing. Maybe there's lots of rich social interaction, there's fighting, you can get gait, posture, all these different information out of this. And I'm going to give you an example of this first. And in this system in my lab, we named it JABS for Jack's Animal Behavior System. And so these are kind of how are the environments in which we can study naturalistic behavior or semi-naturalistic. Obviously, we're not going to go out in the field and look at these lab mice. And I also want to present to you kind of a more complicated model of how we think about the problem and why we're using these ethological studies. So I think of the problem in a hierarchical way. So I mentioned, genetics shapes nervous systems. So we have these, genetic circuits that regulate the formation and the function of these neural circuits. These arrows here imply pleiotropy. There is no one genetic circuit for one neural circuit. A single genetic circuit will control multiple neural circuits, and a single neural circuit will control multiple downstream factors. Okay, so these neural circuits are going to give rise to internal states. These internal states are latent, i.e. we cannot directly observe them. So, if you're an anxiety lab, you're interested in studying anxiety, you may use open field, elevated plasmaze, like dark box, and you would get individual behaviors out of that. If you're a social behavior lab or if you're interested in social interaction, you may do your 3-chamber social interaction test or some other types of social interaction test, and you would, you would get behavior 5, 6, 7. These are just arbitrary, obviously. And if you're a sleep circadian lab, you would do wheel running or EEG, EMG, you would get other sorts of measures out of it, right? But really, how anxious an animal is going to control how social that animal is. What type of social interaction that animal has will control the anxiety level of that animal. We study these in kind of these, separate domains, but they're really not that separate. And what I like to convince you of is by studying here all these behaviors simultaneously, we get a much richer evaluation of the mice. So what if using computer vision in a hotel or a home, we could look at anxiety measures, we could study sleep, and we could study social interactions all at the same time. From an engineering perspective, we could think of these just as features, feature 1, 2, 3, and we can then use these features to essentially rederive the internal state of the animal. So we essentially have a derived anxiety index or sleep index or social index. But these features can be recycled. How much an animal interacts with another animal or sleeps is also a measure of the health or frailty of that animal. So we could essentially repurpose these features for other kind of domains of study. And eventually, we on a per animal level, we can use these indices to construct, let's say, a behavioral profile of the animal. We can perturb that behavioral profile with drugs. We can perturb it with genetic perturbations, and then we can, start to link that genetics and altered nervous system with kind of these internal states and the behaviors. So, a key kind of challenge here is how do you deconvolute these arrows, right? Like if one internal state is leading to behavioral changes here and here, we need to start to think about what are the causal mechanisms. And the way I think of the problem is, as a geneticist, we can use genetic perturbations, i.e. Knockouts, variants, disease alleles to kind of perturb and probe these arrows. And if you're a neuroscientist, you can use optogenetics, chemogenetics, kind of neural perturbations, ablation studies, whatever you want to do to start to perturb these, kind of look at the causal links between these domains. And that's how we kind of really build this map. This is the idea, and we need to really build this map. One advantage is if we keep this behavior method constant, i.e. if my lab uses the same method as any other lab, we can layer the data on top of one another, meaning if your lab studies this circuit and my lab studies this circuit, we can work together and look at the effects together. We don't have to all do the same experiments. Okay, any questions around this before I go on to data? Okay. So like many labs, we essentially kind of have built our own system for collecting and analyzing behavior data. We call this Jack's Animal Behavior System. We acquire video data. You may think putting a camera on a computer on a box is pretty straightforward, but when you're operating at the scale that we are, data acquisition and organization is a major challenge. We then need to annotate behavior. What is the animal doing? This requires behavioral expertise. We train classifiers based on these behaviors. These classifiers can be shared with other labs. So these classifiers encode expertise of individual behavior annotators. So if someone in my lab has built a great classifier for behavior one, there's no need for other people to build another classifier for behavior one, as long as they kind of acquire the data in a similar format as us. And then once we have these behavior classifiers, we characterize the behavior, and then we integrate behavior with other behaviors or behavior with other modalities of data. So our setup is very much an open field setup. It's a 2 foot by 2 foot square foot box. Right now, we've kept it fairly vanilla with nothing in it, but the idea in the future is to add different things in there. We can have a discussion around that. And so we're acquiring and operating on data in this apparatus. All the data I'm going to show you today is 1 hour open field. But over the last 2 years, all our data has really been that the majority of the data we've been collecting are 4-day-long continuous recordings of mice in groups of 3. So we watch kind of social behavior in all these mice. So we have essentially, once we have acquired the video, we built essentially an app, a machine learning app for annotating behavior and building classifiers. So this is what's known as active learning system. The idea here is, here's a snippet of frames, and these are all small videos. Here we're training a classifier for walking behavior, and the user has gone in and labeled this part as walking. Oh, sorry, this part is not walking, and this part is walking. This part is not walking. And then the classifier uses this information to just learn. And then the user then goes in and annotates more frames where the classifier is making errors. And then you kind of guide the classifier towards a working solution. So this is a kind of a classic active learning system. Once we have, so I'll skip some of this stuff, but we have a lot of kind of internal built-in, QCs and metric type stuff into here. So once we have these classifiers, what we built is a JABS web app. And in the web app, the user can essentially take the classifier from here, let's say a walking classifier, and upload it. And this then allows other users to download that classifier app, that classifier, and apply it to their own data. The second thing that we can do is underlying this web app are 3 data sets, a JABS 600, a JABS 1200, and a JABS BXD. This work was very much inspired by what's been done here by Rob Williams and colleagues. And the idea here is that we've taken 600 videos across 60 strains, 5 males, 5 female, and those videos are just kind of sitting at the web app. So when someone deposits a classifier, the classifier gets inferred in any one of these 3 data sets. And then you essentially have walking metrics coming out of all these videos. So you would know the amount of walking, the size of the bouts of walking, the number of bouts of walking across these 600 animals. This instantly allows you to do genetics. You can calculate heritability from these. And you can also do GWAS analysis on this. So just by annotating the behavior and uploading it, you share it with the community, and you get this wonderful genetic information back that you can work with. So that's, this is also part of this JABS application. Yeah, so genetic heritability, we calculate here. Oh, also genetic correlation. So you can look at amount of walking behavior and how it's genetically correlated with, any of the other traits that you pick or other classifiers that have been uploaded to the database. So if walking correlates, with, like, for instance, we did exercise with left turn and right turn, and we just showed that they're different behaviors because you're turning left or right, but really underlying construct is turning, and genetically they're like 90% correlated with one another. So it doesn't really matter if you score left or right turn. You're essentially you're studying the same underlying genetics. I'll skip this. Okay, so then let's get into, so great. So you've got, we've got this whole platform for annotating and behavior and classifying behavior. And what we're interested in doing is kind of taking video data and then splitting it up and segmenting it into behavior 1, behavior 2, so on and so forth. So the field has taken several approaches to this. On the right is probably the more common and more popular approach currently, which is unsupervised learning. There are 3 major methods, maybe 4 now. The oldest is motion mapping from Gordon Berman's lab, and then MoSeq from Bob Dada's lab, and then Eric Itri has B-side, and then there's a new method called BAME, which operates on kind of a recursive neural network type approach. Each one has very different underlying methods, but the idea is in an unsupervised way, it's segmenting the behavior into small motifs. Generally, the critique here is these motifs tend to be very small, meaning 300 milliseconds is the average size of a behavior motif in MoSeq. So you have to make sense of that after the fact if you care about that. Maybe you don't care about that, but that's kind of the challenge with the unsupervised method. The supervised method, the challenge, of course, is you have to do all this labeling on the front end. But once you do that, you know what behavior you're studying. So there's some a priori knowledge that you have to start with for supervised. Once you know you want to go down the supervised route, you can kind of do behavior annotation with abstraction or without abstraction. I'll give you an example without abstraction in the next experiment. So here, we're interested in looking at grooming behavior. So it's a series of small and mid-size and fine motor movements. This is our video right here. This is from Kent Barrage's lab. So the question was if we can have a classifier that takes the raw video, nothing in between, so just input is the video, and classifies the behavior, then we could essentially use that same architecture for any behavior just by swapping out the training data. So we did that. We essentially used a 3D convolutional neural network, and we put in, the input is 16 frames, that's half a second of data. And then we ask it to predict on the 16th frame, is the animal grooming or not grooming? It's a pretty straightforward approach. It works really well. You'll see this goes from red to green when the neural network thinks the mouse is grooming. And the performance here is at human level performance, around 92 to 93% accuracy on a balanced data set. We don't exceed 92, 93% because humans don't agree more than 92, 93% of the time when an animal is grooming. But this works really well. The challenge with this approach is it needs massive amounts of training data. So we have to have a little army of undergrads sitting there labeling videos. But once you have that, it's a perfectly fine network that, that you can use at scale. Let me skip this. Okay, so what can we do with this? That's the fun part, right? So now we've got a classifier. We can classify grooming behavior at scale. Here are 2,500 videos, 2,200 hours of video. Every column here is a different genetic background. Every dot is a 1-hour video. The colors indicate males and females. So you see just the massive amounts of variance in this grooming phenotype. You could also think of this grooming as of high interest to the autism research community because it's a representation of kind of stereotype behaviors. So you could easily think of this as every column being a different drug in a preclinical trial. And you're looking at drugs that enhance or lower the amount of stereotype behavior. And so that's the way I think of this. And I won't go into too much of the biology here, but this is published, so there's lots of interesting things that we found in analysis of grooming behavior in the laboratory mouse. We, of course, are geneticists, so we're able to, as I mentioned, take this data and do genetics with it. We identified over 100 QTLs for grooming behavior using GWAS analysis. And then what we can also do is we can link the genes that we're finding for grooming through phenome-wide association study to traits in humans. So here, for instance, are modules of genes and phenotypes, and the phenotypes here are human phenotypes, like neuroticism, schizophrenia, And the genes are the mouse genes. So it's kind of using the genetics to do cross-species translation. And so we could start to link the underlying genes to human traits for this behavior. Okay, so that was an example of where we just went from raw video to prediction of behavior. The next example is taking a supervised approach with abstraction. And the abstraction we're going to use is key point detection or animal pose. And then we're not going to use classifiers. We're going to use a heuristic rules-based method to get at gait and posture of animals. And the reason we chose gait and posture is that gait and posture underlie just everything. Like if you have muscular disorder, you're going to see gait posture deficit. If you have pretty much most of the brain, any part of the brain that's affected, you're going to see gait and posture deficit. So this is a really important preclinical tool. Maybe this will play. There we go. Okay, so we're taking the raw videos and we're putting these kind of key point detectors on them. We have 12 key points. The challenge here was dealing with the diversity of the mice. We have black mice, white mice, mice with no fur. But this was a handleable challenge in the sense we had to just label more data and train our, so this is the nude mouse, train our algorithm more. The harder part was going from key points to actually stride and posture. And this sent us back to literature from the 1970s and 80s. These are classic Hildebrand plots. So a limb is making contact with the ground. So this was a strike phase and a swing phase of a stride. So Hildebrand kind of put all strides in this percent or stride time. And then he measured when limbs were making contact with the ground. And here, for instance, we've done this. So every time the velocity of this orange goes to 0 is a strike with the ground. And so the mouse, the right leg strikes here, 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 so on, and animal makes a turn. The left is kind of antiphasic to the right. And then this green is a point on the body, and that point is usually constant. So by looking at the velocities, we're able to extract this information. The second thing that you get in terms of this approach is the overall posture of the animal. So these are the same data. These are the key points, the nose, the base of the tail. And this is kind of going up, it'll come back down, and it'll end up in the middle. So the mice are not just walking straight, they have this kind of swagger about them. And looking at that swagger, we understand the posture of the animal in a stride cycle. The other part of this is because we're allowing the mouse to freely behave, we get lots of repeated measures. So every dot here is a stride. So we can very precisely measure the phenotype. So this is the posture so we're looking at a point on the tip of the tail over the stride cycle, and that point goes up, comes back down and ends up in the middle. The nose point is antiphasic to the tail. And right away, we knew this was important. So these are 10 different mouse strains. And you see the diversity of this, this kind of posture movement. And the fact that this varies across genetics tells us that this is a highly heritable strain. And in fact, when we did our GWAS analysis, the most heritable component of mouse movement was the posture. It's not what people have been looking for 50 years, such as stride length, step length. It's really the posture of the animal that's the most heritable. So we validated this using models coming out of the repository, a Rett syndrome model, a ALS model. Just going to show you data with Down syndrome model. In a Down syndrome model, Down syndrome patients have motor coordination deficits. So you look for that in the mouse using an ink blot analysis. So this is data from Muriel Davison at JAX back in the 90s. And if you look at the Down model, it's slightly erratic and not as regular as the control mice. We looked at the same model in our open field assay, and this is that tail tip lateral displacement measure. And you see the controls at 3 different speeds and the Down model at the analogous 3 different speeds. It's night and day. they're completely like antiphasic to one another. So it's like we have this great, really strong phenotype in posture and movement of the animal in this model. And so I'm going to go back to this idea of studying genetic circuits, neural circuits, and these kind of internal states, right? So here's a genetic circuit that's perturbed, and it's leading somehow to cognitive deficits, which is the hallmark that we want to work on in the down cases. But here in the mouse, we're also seeing motor deficits. So in the preclinical space, cognitive deficits are really hard to work on. Like to do a drug discovery project in a studying cognition in an animal model would be really challenging. But here I'm showing you a very simple assay where you just let the animal move around and you can get really fine-grained motor movement of the animal that's different. So the question is, if we're studying the same construct, could a drug that affects motor also affect cognitive deficits? And could the motor deficit be used as a screening tool for the more complicated assay? Okay. So that's kind of one idea. And we can think of this as like a phenologue, like a phenotypic homologue. I'll skip the genetics since we're running short on time. Okay. Okay, so quickly, just a couple of other approaches, because the supervised approach is broad. So I talked about no abstraction. I talked about a key point abstraction. I'm going to tell you just quickly about segmentation-based abstraction. So this is an abstraction where you start with raw video. We're using a neural network, and we're going to segment the mouse into a mouse or not mouse. So everything here white is not mouse, and this is the mouse. So you essentially have a blob that describes the shape of the animal. And from this, we were interested in looking at sleep. So could we predict wake, non-REM, wake, yeah, this is mislabeled. So wake, REM, non-REM, or non-REM, REM from just the image. And we actually can. The types of features that we use are essentially spectral features. So if you're a signal processing person, you recognize these. So it's like this is the image moment, and then we can operate on the raw signal. We can operate on the FFT, like fast Fourier transform of the raw signal. We could use autocorrelation to extract essentially features that describe the raw signal. And we use these features to train a model, and the model, the ground truth of the model is coming from human annotated EEG, EMG data, okay? So I don't have time to go too much into this, but this actually works really well. And in fact, like in many cases we found you don't actually need key points. You can operate just on the segmentation, and you can get really fine-grained information about what the mouse is doing and the behavior it's carrying out. Okay, so we have these different methods. In the end, you may not really care about individual behaviors. You may just want to extract information like how much pain is the mouse in, what's the biological age of the animal. So here we worked with the Shock Center, and Gary Churchill came to us and said, well, frailty is the major, is the best indicator of mortality in humans. The frailty index is widely used to measure biological age in humans. In mice, they've got a frailty index also. It's essentially humans, frailty index is hundreds of measures coming out of electronic health records, things like, objective measures like blood glucose levels, BMI, all these kind of other things. In mice, you have 27 items, and the human is going in and quickly evaluating each one of these 27 items and saying it's affected, 1, maybe affected, 0.5, and not affected, 0. And then in the end, you just aggregate all these numbers and you get a frailty status of the mouse. So the question here is, could we use behavior to study frailty in these animals? And is the behavior, is it enough to really tell us, to let's say, replace or augment the traditional frailty assay? So here what we're going to do is we're going to take this series of video, time series data, and we're going to extract as many behaviors as we can out of them. And then we're essentially going to aggregate all these behaviors to predict a frailty level. We're not going to look at one behavior. We're going to look at as many behaviors as we can figure out. So we don't care if it's key point based, if it's no abstraction. We're just going to have these individual behaviors as features, as I was mentioning before. So right away, like we knew that behavior encodes information about the biological age of the animal. So this is a 2-month-old animal. You don't need this label here. You can tell a nice limber animal from a not so limber animal and a older animal. The question is this enough to tell us the frailty status of the animal? And the way we did this is we essentially generated one of the largest video frailty data in the block 6s. So these are 643 data points. So these are animals that were tested for manual frailty by the shock center and the experts there. We tested all these mice in an open field assay, and then we extracted all those measures that you saw, the gait, the posture, the grooming, all these different things. We even engineered a bunch of features. And then we used these features to essentially train a model to predict the manual, the frailty status. And I'm not going to show you the exact data. I'll just kind of quickly point out that what we find is that this works really well. And in fact, we can, the error here is 1 out of 27 items, or 2.5 items out of 27. So within a very narrow interval, we can tell you what the biological age of the animal is just by examining it using our methods for 1 hour in the open field. Yeah, so just as a summary of this kind of part of looking at single animal behavior, we're interested in this problem of behavior extraction. We did several different approaches. I showed you an approach of classifying raw video. I showed you an approach of classifying blobs, essentially segmentation. I showed you an approach of classifying based on pose with rules, which are the gait and the posture rules. We can also kind of use these kind of methods to classify things like licking or shaking type behavior, and we can study pain behaviors. I didn't really talk about this. And then we can kind of merge all these features to predict things such as frailty. The idea is just, many ways to kind of skin a cat in the open field. We're really good at this. I think the challenge is the model itself. So, this is a great space for collaboration between labs like mine and other labs that may be experts in specific models. And it's really that data that is needed to kind of train these algorithms. That's the key. Okay, so I'm going to switch from the hotel approach, which is our open field that I showed you, the home approach. So while we were doing, all these for the past 4 or 5 years, I've been working very closely with JAX, the business side of JAX, which is still a nonprofit, and they've taken our algorithms and placed it inside a home cage environment. And so the goal here is to empower the biomedical research community through this digital in vivo initiative. And this is leveraging machine learning and AI methods to understand rodent behavior in a 24/7 home cage environment. We've essentially automated the hard part here of data collection, storage, and we have some analysis tools, but the analysis tools need to be built up a lot more. And this is, I think, needs to be a more community effort. Yeah, so let me show you. The approach is similar, right? So we have essentially portals for setting up your study design. Then we have local data acquisition hardware, which is custom hardware that have cameras in it. All the video data gets uploaded to the cloud where machine learning kind of foundational metrics are applied and extracted. There's display and visualization of this data. And then finally, no one size fits all is going to work for everyone. So we've invested a lot in kind of these advanced behavior quantification workbench. So the idea here is that you can very specifically start to ask the questions that you want to ask, but the data is residing in a really organized way in the cloud, hooked up to lots of CPUs and GPUs, so you can operate at scale. Okay, so this is the digital cage. This is a rat-sized cage, so it meets like European standards. So it's a pretty big footprint. Well, not huge, but a much bigger footprint than a normal mouse house that you're probably used to. This is the business end of the whole apparatus. So there are 2 cameras embedded up here, along with a Jetson single board computer and lights and all infrared and visible lighting. So everything is here. This is a self-contained unit. It's got like a HEPA filter, so the air is sterile. That comes in here. From the inside, it looks like this. It's just a top-down camera. We've tried to eliminate as much of the blind spot as possible. So the food is over here, the water is over here. There's space here. There's a running wheel. I don't know if you can see it, but there's a running wheel here. This is optional. We can remove it if you like. This is what it looks like when interesting things are happening. So this is in the dark, and you can see there are 3 mice. One mouse is eating, one is grooming. This may look dim to you, but there's a lot of information here that the computer can use, and this works really well. It's highly scalable. So this is one of our DAX rooms at Jackson Labs. We have 4 of these shelving units, and it's a scalable system. We've been using it for the past year and a half. These are slightly older numbers, but yeah, I mean, we have multiple sites, including some pharma companies that are using it. We've collected, trillions of frames of data. It's just some sampling. I think this is supposed to be a video, but it may not play, but environments. But the idea here is, it doesn't matter where you are, which part of the world, all the data gets uploaded to a cloud service, which then allows you to do experiments. So essentially, I can run an experiment 1,000 miles away very easily, as long as there's a person there to place the mouse in a cage. Okay, so the interesting part for us, what do you do with all this data? So this is the machine learning pipeline, this foundational metric right here. So a video goes in, and this is HD video, and essentially we have this, so this is a design and engineering challenge also, which is when you're operating at the scale, you need to really focus on efficiency. So this is a mobile net backbone that is designed in kind of a Hydra network. So this is our feature extractor right here. And then the features kind of go down 4 different heads. Head 1 is object detection. So essentially putting these yellow bounding boxes on the mouse. So its job is to just find a mouse and put a box on it. The head 2 is a segmentation head. So this is an encoder-decoder network that's going to do what I showed you earlier, which is a mouse, not mouse. So here's an identification of a mouse. Object 3, head 3 is a key point head. So it's putting 5 points. So the key points we're using is nose, ears, the middle of the back, and the base of the tail. That's it. We do the tip of the tail, but I don't know, tip is always challenging. It gets buried in the bedding. So we have a key points head, and then we have kind of what we call offsets head to do instancing to tell which mouse is what. And the end result is an image like this, where you have kind of all those metrics overlaid onto every image. So this neural network is going frame by frame on millions of frames collected over the course of an experiment and doing this in all these frames. We can then take this data and extract metrics. So here is a night dark, every night dark 24 hours. So this is over 30 days of data of these mice in a cage. So this is the top camera. This is the side camera view. And if I take this little part here and blow it up down here, you see this nice diurnal activity rhythms. We can look at average movement of the mice. We can look at wheel occupancy. We look at water dispenser or food occupancy. But the idea is this allows you to view the data. You can share your study with whoever you like. You can annotate things. So for instance, hey, Bob, looks like that mouse is not eating as well or hasn't done something or is doing something interesting. You can tag them right here. So you have these kind of social media hashtagging type approach to the data. And then finally, we have this kind of workbench that I mentioned, and on the surface, it looks like there's lots of just these notebooks. But the idea is because all the notebooks are organized, when I ask a question, my code is available for the next person to use on their own data. And the other, I think, important part is all the video is sitting in one nice organized containers. or location, so you can really access them very easily. So this is a scalable platform for with ML tooling, gives you direct access to the videos and all the metrics. It has all the commonly used training, testing, and development examples. It essentially is a robust infrastructure that you can use with your own kind of questions. I'll just give you one example of an experiment that we've done recently. So this is home cage activity of 8 different strains. So we have AJ, BALBs, BTBR, C3H, Black Sixes, C57Ls, Castaneus, and FBB. This is 2 weeks of continuous monitoring. And there are 9 mice. So there are 3 groups of 3 mice. So actually, you're not going to be able to see it, but there's actually 3 lines underlying each one of these panels. And yeah, I mean, you can see just how consistent that data is across these males and females. So this is kind of looking at data across all 2 weeks for the males and females. This is data. What we've done is essentially a waveform data. So we've collapsed all 14 days into one data set here. And, you know, I think for the aficionados, AJs are the ones that just go sit in the corner. They're little balls of dough, as I've heard them being described, but you can clearly see AJs are very robust activity rhythms. And FBBs, I was surprised, like, you know, FBBs in our open field, 1 hour assay look fairly active, but they really are very, their circadian organization or their diurnal rhythms are highly disorganized. The activity at night and day, you can kind of see them here. This is night and this is day. There's not that much of an amplitude compared to something like a Black 6 or AJ. So yeah, so I think, you know, the nice thing here is now that we've generated this data set, we can share this with anyone and they could ask very specific questions using this data. And what we hope to do is actually build up this repertoire of strains to, you know, dozens, if not hundreds of mouse strains and share those videos with the community. So there's a website, diva.bio. Right now, it's the major kind of part of this whole group are pharma companies. There's 5 or 6 if you include JAX. But the idea is the major kind of users right now are the developers. These are the engineers, the computer scientists, and the groups that are kind of really investing in this. And then ultimately, we want to get it to the researchers like me and you hopefully are going to develop new assays to get past the forced swim test and the tail suspension test and do more interesting home-based and hotel-based tests. And then we imagine the largest group will be just users who are not really interested in developing assays or technology. They're just using the latest and the greatest methods to run their studies. But we see this ecosystem as kind of a really evolving ecosystem. And this will be fully kind of available next year sometime. That's the expected timeline right now. Okay, so I kind of talked about our homes, the first half, our hotels, the second half of the data. And I kind of started with this question of developing better preclinical animal models to solve this problem of treatment of neuropsychiatric disorders and other disorders. And I've shown you lots of, where we think the technology is really advanced and we're in a good spot, but there actually is a huge amount of work. We're still in this infancy of this technology. And the way I see kind of the major problem, the challenges in the future are things such as what I call ethological tasks. So we're observing the animals, they're doing naturalistic things, but if you want to challenge, you want to have a probe for a specific neural or genetic circuit, we still may need to give them very specific tasks. Maybe these are tasks such as finding their way to food in a maze, or maybe it is, some other types of tasks. And so I see kind of development of ethological tasks to probe circuits as a key part of this ethological approach. We need to essentially build a behavioral space based on genetics and pharmacology. So I showed you data from strains, I showed you grooming behavior, gait posture, that list in my kind of the hierarchical approach, that feature list is still very rudimentary. I think we can have thousands of features that describe the behavior of the animal, and we need to really build this behavioral space. I think democratization of technology is still a major challenge. Most labs are still not equipped to start using these methods. I'm sure, if you're delved into this, you'll encounter this. So this has to be, the barrier to entry has to be lowered. And then finally, even if this happens and everyone jumps into the field, there's still, I would say, an infrastructure challenge. Video data is very large. You need lots of GPUs and CPUs and TPUs to operate on that data. So this, when we found the NIH resources that come from, or NSF resources are not adequate for either storage or for operating on this much data. So these are kind of challenges for the future that needs to be tackled by the community. And with that, I'll just thank the funding agencies and the folks that did the work, and I'll take questions. Thanks. Yeah.
Congratulations on groundbreaking. That is fantastic.
Thanks.
I think you mentioned that these pathological tests, like for example, gait behavior, right? Mm-hmm. It's pretty easy to probably understand this. But when it comes I do visual behavior, mm-hmm. It's a lot harder to kind of discriminate whether the mouse is able to see a particular task or not.
Yeah.
And it varies from day to day.
Yep.
And especially after challenging with a drug or anything.
Yeah.
So
yeah.
I think that's probably the biggest challenge.
Yeah, I totally agree, right? Especially what I didn't show you is most of the data that we're collecting is completely in the dark. We've gone out of our way to make sure the mice have no light input at all. And we have actually tasks that I didn't show you that are maze tasks. So we have complex mazes and the mouse has to navigate that. And they do that task really well in complete darkness. So as a vision researcher, I would ask, what good is the eye to a nocturnal animal? But that's besides the point. But yeah, I mean, I had a conversation with some internal researchers at National Eye Institute exactly on this topic. What are the types of tasks you may use for visual systems, visual discrimination? They talked about kind of this task where you had I think you throw a live cricket into an arena and the mouse will literally hunt that cricket, and they're really good at hunting that cricket. And yeah, I never thought of mouse as a predatory till I saw some of these videos of them hunting crickets. Yeah, so this is where I think there has to be collaboration between labs like mine that are more tech dev and labs like yours that are more on the application end to really determine what is the right behavior, and more importantly, what is the right model to validate that behavior, right? And as you know, vision is not just one thing. Vision is many different things. And so you have to really start thinking about more carefully about that. And in certain tasks, I would say you don't need it. If you're doing ERG testing, and if you're happy with that, and that's a gold standard test. And if that works for you, yeah, and if you find that scalable, that's fine.
We supplement or complement the studies with different studies.
Yep.
And if everything aligns in the direction that we think, right? So it's great.
Yeah.
But oftentimes, we have two distinct data points. So ERG shows fantastic outcome.
Yeah.
And the optokinetic measurements are totally different.
Yeah.
So
yeah,
that's where the question comes,
yeah.
Which one is correct?
Yeah, that's a great point too. Yeah. There's one there, and then go ahead.
So, showing mostly adult animals, do you have any, how young kids?
How young? Yeah. So we've gone 3 weeks, 4 weeks around that. And the tracking gets challenging when you have 3-4-week-old mice. You don't see the limbs as well. But we're working on a project with play behavior, and we see very robust play behavior in mice at 3-4 weeks. Yeah.
Great talk, really exciting technology. You mentioned at the beginning the limitations, and I totally agree with some of the tests for depression and anxiety, behavioral tests that are currently being used. Do you have thoughts as to how you would use this approach to develop better measures related to anxiety and depression, and also the validation that's been done for the tests we currently use are based on a limited class of drugs. They work well to predict the behavior of those classes of drugs. So you have thoughts of validate if you develop new methods that would have a broader class of drugs.
So I think that's a key kind of challenge in the field. So if we can agree on this part, let's say we observe animals for 4 continuous days and we observe things like eating, drinking, socializing, sleeping, social interactions, everything, and how much they're defecating, where they're nesting, all these different things in our open field. And that's what we actually do now. Like that's what we do for 4 straight days. Then what I would do is I would administer the mouse an antidepressant, perhaps at a sub-efficacious dose, at an efficacious dose, and a maybe toxic dose. And I would start to see what changes here. And I would do that with not just one drug, but hopefully like all the compounds that are approved for the treatment of depression. And I will build like a behavioral space for that drug. And ideally, I'd like to do that not just in the Black 6Js, but in all these BXDs, so you have genetic diversity as part of the mix. And then once we have this kind of multidimensional matrix of data, I could tell you very accurately what is the signature of an effective drug and what is the signature of subclass of this type of drug. So that's kind of one approach is this pharmacological approach for drug discovery. A second approach would be, let's put in, some like a DREAD type system and turn off or down-modulate dopaminergic signaling, and or in your case like NPY type signaling, and start to look at anxiety phenotypes and things like that. So I think those are the 2 approaches that can be taken. A third approach would be more at this level, but this I think will get messier. But if we have specific models of depression where you have single gene knockouts or single alleles or mutations that lead to a depression-like phenotype, and the community buys it, then I think we can characterize that and then look at what's different here. But that's the kind of work that has to be done. The power of it is if your lab and my lab and all other labs kind of adopt this system, then, like one lab could work on anxiety, another lab could work on depression, another lab could work on sleep mutants and drugs, and we could all kind of share this data. I mean, this is what the magic of Rob did, right? Like, I mean, he really figured out a way where the community could use the same genetic tools and then aggregate all that data across scales.
Your data on APV mice was very interesting, but those are APV mice is carrying RD1 mutation.
Yeah, exactly.
Essentially blind.
Yeah.
So definitely that is affecting their circadian system. So as you said, you are trying to developing that genetic space or pharmacological space.
Yes.
Also, some kind of phenotypic space of the lines you know already. So that probably will help in streamlining some of these assays. Yeah,
I completely agree. So lots of lab strains, including C3Hs here, go blind as they get older. I will point out that if you're blind, does not mean you don't entrain, because melanopsin cells are the ones that are sensors for light. But still, yeah, that's absolutely the case, yeah.
What about unsupervised approaches? Could those be also a way to share data? The depressed model of a mouse being conditioned in an unsupervised model without actually assigning features, let's say.
Yeah, I think that's a very valid approach. The current methods of unsupervised methods don't have a shared embedding space. So if you do experiment 1 with 10 mice and you do experiment 2 with 10 different mice, just the way the embedding space works out is you'll end up with different number of clusters, different locations. So you have to then kind of go start to do 20 mice combined and do another analysis. So that's one thing.
You need the comment better than what you run across the video data.
Exactly.
That would
Yeah, there are newer methods that are trying to deal with exactly that problem. The second issue, I think, and again, this may not matter, but as a behaviorist, I want to know what the mouse is doing. But you may not care. Like if you're looking at drug effect, you just need to know that motif 10 is changing. Maybe motif 10 is just the mouse doing this, and that's it. So yeah, so these are kind of the things. And then I think there's also space for combined approaches where you used unsupervised methods to get cheap labels for supervised. So you do some sort of semi-supervised approach, because the power of the supervised is once you have that classifier, you know exactly what you're getting. And you don't have to worry about space or anything like that. You can apply to 10 different videos and know exactly what you're getting. So, we could like have a hybrid approach also. Yeah. Yeah.
So can the individual behavior predictors used in the video of 3 minutes?
Yeah, it can. Yeah. So so the way we are doing the single animal work is it's egocentric. So it's operating on a single animal, and the as long as the mice are separated enough so the key points don't get mixed up between mice, we're able to like apply the single animal classifiers to multiple animals.
Segment image.
Yeah, so the top down versus the bottom up type of approach. So we stride with the bottom up because sorry, is it the bottom up or the top down? One where you detect all key points and then you assign different identities to each key point, and that one doesn't work very well. So then we're doing the other approach of finding mice and then doing key points on each one of the mouse. It increases the workload quite a bit because now for every frame you're doing at least 3 sets of inferences. But it's more accurate.
So for that original abstraction.
Sorry, the what?
The one without abstraction.
Yeah.
The first video you showed.
Yeah, so for almost everything now we're doing is some sort of abstraction. It's just much more efficient. Like we cannot afford to label 24 hours of video data for every behavior. It's much more accurate, but the cost is very high in terms of labeling. So we definitely we're using some sort of abstraction, either key points or segmentation as an abstraction. Okay. All right, thank you very much.